Hello everyone, I'm Dr. Atira from Biotechnology Research Institute, University, Malaysia, Sabah. In this video, we will discuss on a topic entitled Natural Products from Microorganisms, Part 1. For this topic, we have 5 sections, but for this video, I will only cover 2 sections, which are the introduction of microbes, particularly from bacteria, archaea, fungi, and virus, and the special features of microbial metabolites. As an overview, we will learn about microbes. We will focus on bacteria, archaea, fungi, and viruses. Next, the characteristics of microbes, followed by the definition of microbial metabolites and the special features of microbial metabolites. Let's start with microbes. Do you know what is a microbe? So, what are microbes? Microbes are organisms that can only be seen using a microscope. They are widespread in nature and are beneficial to human life, but some can cause serious diseases that are harmful to humans. They exist as unicellular, multicellular, or cell clusters. Microbes can be divided into 6 major types: bacteria, archaea, fungi, protozoa, algae, and viruses. In this video, we will focus on 4 types, which are the bacteria, archaea, fungi, and viruses. Let's start with bacteria. I'm pretty sure many of you have heard the word bacteria. Bacteria are unicellular organisms. A bacteria consists of a single cell. This means all life processes such as reproduction, feeding, and excretion occur in one cell. Bacteria exist in 4 major shapes. We have the bacillus, the rod shape, coccus, spherical shape, spirilla, spiral shape, and vibrio, curved shape. Example on the left side, you will see an image of E. coli, a gram-negative rod-shaped bacteria. Bacteria can be classified as either gram-positive or gram-negative based on staining method. Bacteria are divided based on their response to oxygen into the following groups. We have the aerobic, living in the presence of oxygen, anaerobic, living without oxygen, and facultative anaerobes can live in both environments. Bacteria are also classified as heterotrophs, whereby they obtain their energy by consuming other organisms. Autotrophs, they make their own food by using the energy of sunlight or chemical reactions, and saprophytes, they use decaying life forms as a source of energy. Now let's look at the bacterial structure. Bacteria are prokaryotes. They are lacking of well-defined nuclei and membrane-bound organelles. Let's look at the cytoplasm. The cytoplasm or protoplasm is the bacterial cell where the functions for cell growth, metabolism, and replication are carried out. It is a gel-like matrix composed of water, enzymes, nutrients, waste, and gases, and contains cell structures such as ribosomes, chromosome, and plasmid. The nucleoid. The nucleus is a region of cytoplasm where the chromosomal DNA is located. It contains all or most of the genetic material. It is not a membrane-bound nucleus, but simply an area of the cytoplasm where the strands of DNA are found. The cytoplasmic membrane, or also known as the plasma membrane, it is a layer of phospholipids and proteins regulating the flow of materials in and out of the cell. It acts as a barrier that allows selective interaction with their environment and constantly adapting to different conditions. The cell wall is composed of peptidoglycan, a polysaccharide molecule. The wall gives the cell its shape and surrounds the plasma membrane, protecting it from the environment. The cell wall is also responsible for keeping the cell from bursting, especially when there are large differences in osmotic pressure between the cytoplasm and the environment. The capsule is a third protective covering made up of polysaccharides. Capsules keep the bacteria from drying out and protect from phagocytosis. The flagellum is a long, thin, whip-like structure that provides a mean of locomotion for bacteria. The flagella beats in a propeller-like motion to help the bacteria move towards the nutrients and even away from toxic chemicals. Pilus is a small hair-like projections emerging from the outside cell surface. This assists the bacteria in attaching to other cells and surfaces. Now let's look on the bacterial shape. Bacteria come in many shapes and sizes. The basic bacterial shapes are bacillus, coccus, spiral, and vibrio. Example of rod shape is Bacillus anthracis, the agent of anthrax. Example of coccus-shaped bacteria is the Staphylococcus aureus, a type of bacteria found on human skin. Example of spiral-shaped bacteria is the Leptospira interrogans that causes leptospirosis in human. An example of vibrio-shaped bacteria is the Vibrio cholera, which causes cholera in human. Now let's have a look on archaea. The archaea is a single-cell prokaryotic organisms that have distinct molecular characteristics separating them from bacteria in eukaryote. They were firstly discovered and described in extreme environments such as the hydrothermal vents and terrestrial hot springs. Many of them can be found in a diverse range of highly saline, acidic, and anaerobic environments. These are the example of archaea that live in extreme environments. The sulfolobus species A20 is an example of an acidophile and thermophile. It can live in very acidic, low pH condition, pH 2 to 3, and thrive in high temperatures between 75 degree to 80 degree. The Pyrolobus femeri is an example of anaerobic and hyperthermophiles. It can live in the absence of free oxygen and thrive in extreme high temperature between 95 degree to 113 degree. The Thermococcus gamma tolerans is an example of thermophiles. It can thrive in high temperatures between 55 degree to 95 degree. It is the strongest and most gamma ray radiation resistant organism known to exist. The Methanococcites burtoni is an example of psychrophile. It can live in an extreme low temperature environment as low as 1 degree. Now let's move on to fungi. Fungi are eukaryotic organisms. They may exist either unicellular or multicellular. Some are microscopic in size, while others form much larger structures, such as the mushrooms. Fungi, however, do not contain chlorophyll. They absorb and dissolve nutrients. They can be found in the air, on plants, and even in water. They can cause a number of plant and animal diseases, such as in human, ringworm and athlete's foot diseases, while in plants, leaf, root, and stem rot diseases. The fungal structure consists of three major parts. Sporangia is an enclosure in which spores are formed. It can be composed of a single cell or can be multicellular. Sporangia 4 is a specialized hyphae bearing sporangia. It is a filamentous stalk on which a sporangia forms. The rhizoid is the subsurface hyphae that are specialized for food absorption. They grow downwards and anchor the fungus. They release digestive enzyme and absorb digested organic material. Next is the virus. Do you know the word virus is a Latin word referring to poison? Viruses are obligate intracellular parasites. They depend on the biochemical machinery of the host cell for replication. Viruses are different from bacteria by being filterable agent. They are able to pass through a very small filter that cannot pass through by bacteria and parasites. Viruses are not considered living organisms because they cannot reproduce by themselves without a host, and they do not have cells. However, viruses contain nucleic acid genomes, genetic variation, and even they can evolve. Hence, it is questionable if viruses are considered as living organisms. So, how are viruses different from bacteria? Well, bacteria are single-celled and considered as living organisms. Bacteria do not depend on a host cell to reproduce. Bacterial and viral infections are treated very differently. Antibiotics only work in bacteria. The diameter of a typical virus is about 20 to 300 nanometer, while the average size of most bacteria is between 0.2 and 2 micrometer. These are the characteristics of viruses. Viruses are acellular infectious agents, meaning they do not have a cellular structure. They don't have components of cells such as organelles, ribosomes, and a plasma membrane. Viruses are obligate intracellular parasites, meaning they cannot reproduce outside their host cell. They are entirely reliant on intracellular resources such as humans as their hosts. Viruses possess either DNA or RNA, but never both. Replication is directed by viral nucleic acid within a cell. They are considered as non-living things. They do not divide by binary fission or mitosis like bacteria and other living organisms. Viruses depend entirely on host cell ribosomes, enzymes, and nutrients for protein production. Viruses lack of genes and enzymes necessary for energy production. They don't have mitochondria to generate their own energy. They have to derive their energy and all other metabolic functions from the host cell. Several viruses can cross the placenta and cause developmental disturbance, such as the Zika virus. A complete virus particle is called a virion. The components of virions consist of nucleic acids, capsids, and envelopes. The nucleic acid carries the virus genome, the collection of genes, and may consist of either DNA or RNA, but never both. The protein capsids are made up of many protein subunits called the capsimers. Protein capsids provide protection for the nucleic acid and may contain enzymes that allow the virus to enter its host cell. The envelope consists of proteins, glycoproteins, and host lipids derived from host membrane. They may help viruses to avoid the host immune system. So now we have learned about bacteria, fungi, archaea, and viruses. A variety of organisms such as bacteria and fungi produce secondary metabolites, also known as natural products. Microbial metabolites are chemical or carbon compounds isolated from microorganisms. These secondary metabolites may have antimicrobial, anti-tumor, antiviral, and anti-cancer properties. Antibiotics and other bioactive microbial metabolites are produced by both prokaryotes and eukaryotes. Among prokaryotes are the Bacillus, Streptomyces, Mycobacteria, and Pseudomonas species that produce the highest amounts of metabolites. Now let's have a look on the special features of microbial metabolites. The most important characteristics of the bioactive microbial metabolites are their microbial origin, their interaction with the environment, and their unique chemical structures. In general, natural products, including the microbial metabolites, may be practically utilized in three different ways. First is by applying the natural product directly in the medicine, agriculture, or in any other fields. Secondly, is by using a starting material for subsequent chemical or microbiological modification. Next, they can be used as lead compounds for chemical synthesis of new analogs or as template in the rational drug design studies. Alright, so we have covered all the listed topics, and if you have any question, you can always email me. Thank you.